What's up everyone, welcome back to another video on our channel, the Spin Dog Cytokinetics. Today we'll be going over the process of meiosis, but before we start, make sure to subscribe and smash the like button. Anyway, let's begin. The process of meiosis starts with interphase, exclusively preceding meiosis 1, divided into 3 different stages, G1, S, and G2. In the G1 phase, the cell grows. In the S phase, the 6 chromosomes, each with a centromere, duplicate into 2 scissor chromatids per chromosome, resulting in 6 homologous pairs of scissor chromatids and 12 chromatids in the cell. After interphase, the first half of meiosis, meiosis 1, occurs. Before meiosis 1, the cell is a diploid cell, meaning there are 2 sets of complete chromosomes, 1 from each parent. Beginning with the first stage of this process, prophase 1. One scissor chromatid from one pair will cross over genetic material with another scissor chromatid from another pair from the other parent, meaning genes from every 2 non-scissor chromatids switch, increasing genetic variety. This happens 3 times simultaneously. Then in metaphase 1, the 3 homologous pairs of scissor chromatids line up horizontally in the middle of the cell, and in anaphase 1, the 3 pairs get pulled to the opposite ends of the cell with the scissor chromatids still intact. Finally, in telophase 1, the process of cytokinesis divides the cytoplasm equally into the 2 haploid cells resulted from anaphase 1 and gets ready for meiosis 2. Meiosis 2 entails the same 4 stages with slight variation. Before meiosis 2, the 2 cells are haploid cells, meaning there's 1 set of complete chromosomes in each cell. Comparing to prophase 1, the 3 chromosomes from each of the 2 cells in prophase 2 do not cross over. In metaphase 2, chromosomes line up in the middle of the cell, and in anaphase 2, the sister chromatids from each chromosome get separated to the opposite end of the cell. Then finally, in telophase 2, cytokinesis occurs again in the almost formed 4 haploid cells, resulting in 4 gamete cells for the purpose of sexual reproduction, and the formation of cells with homologous pairs, aka 2 chromosomes, 1 from each parent containing the same type of genes after conception. The process of meiosis confirms Mendel's theory through the law of segregation and the law of independent assortment. In telophase 1, the diploid cell splits into 2 haploid cells, which demonstrates Mendel's law of segregation, stating that each gamete has 1 set of the parent's genes. Mendel's law of independent assortment also parallels the gene's unpredictable separation pattern during anaphase 1 and 2, increasing genetic variation. What Mendel missed, however, is that meiosis results in 4 gametes, not 2. If we wish to fully represent meiosis using Punnett squares, it should be done in 4x4 instead of the 2x2 format proposed by Mendel. But since the ratios are proportional, Mendel's theory still functions as a valid trait calculation and prediction.